Your thoughts might be filtered through microscopic brain fluid errors. Imagine your consciousness is not a direct broadcast, but a river system constantly clearing itself. If that filtration, the glymphatic clearance process, gets sluggish with age or stress, noise builds up in the interstitial space between cells. This sludge fundamentally changes how signals are received and perceived. It is not just tired neurons, it is distorted signal processing. A 2023 Nature study showed compromised cerebrospinal fluid dynamics directly impacts waste removal efficiency within glial compartments. Think of astrocytes as microscopic water regulators for your mind. When they fail to manage this internal plumbing, the clear view of reality, the integrated awareness you crave, becomes blurred by functional static. Think about the brain not as an electrical circuit, but as a sprawling metropolis that needs perfect structural integrity. Beyond just waste removal, there is this complex architectural scaffolding, the extracellular matrix, that actively holds conscious experience together. Researchers have consistently found that glial support cells are key to maintaining this delicate local environment, essentially shaping how different parts of your mind talk to each other. This homeostatic balancing act allows for a kind of cognitive coherence we might call integration. It is the physical purity of the perivascular space that permits true non-duality, the seamless blending of who you are and what you perceive around you. But what if that structural integrity we spoke about was inherently fragile? What if the interstitial space itself creates optical illusions for your brain? We are talking about the veil of Maya, where perceived reality is filtered through microscopic fluid dynamics. Astrocytes are not just regulators. They are dynamic signal sculptors, controlling lactate shuttling and glymphatic clearance on a subcell level. Preprints detailing astrocytic signaling roles in plasticity show how subtle failures here introduce noise into neuronal firing patterns. That internal static does not just degrade function, it warps perception itself, creating a false clarity or distorted coherence. Which means something nobody expected for the subjective self. Your reality might be an elegant metabolic error. But if we can stabilize this microscopic environment, what new forms of integrated awareness become possible? But if that internal static is warping reality, can we even control it? The stakes are higher than mere perception failure. This speaks to how complexity itself is built locally. Imagine your brain not as a monolithic computer, but as an intricate clockwork mechanism held together by molecular architecture, the ECM scaffolding and ion balance across the perivascular space. This structural integrity is not passive. Researchers have consistently found that glial support cells actively govern these microenvironments, acting like master builders, ensuring every component fits perfectly to generate complex patterns of thought. It is a precise chemical orchestration required for integrated awareness. But while this view suggests stability is key, some studies contradict this notion entirely, positing that localized stress may actually drive higher-order integration. How much optimization do we need before the system achieves something genuinely new? The final frontier is not just balance. It is metabolic efficiency. It is about optimizing nutrient and waste exchange kinetics in those tiny spaces around vessels. When these conditions hit peak purity, the brain does not operate like a siloed set of modules anymore. Think of your cortex as an orchestra where every instrument plays perfectly integrated harmony instead of separate solos. Functional imaging data shows that when cerebral perfusion meets this optimal metabolic rhythm, sensory input and executive function demonstrate maximal integration without any functional segregation artifacts whatsoever. The state is the closest we get to true non-duality in biology, the perfect blending of observer and observed. But how do our bodies even begin to hit that exact sweet spot? The signaling pathways involved are far more intricate than mere fluid dynamics suggest. They require a precise cascade. The precise cascade we need suggests that stability alone is not enough to explain integrated consciousness. We have been viewing the brain as a system of pipes and circuits. But what if those interstitial spaces are actually dynamic, microscopic lenses? The idea surfaces that fluid exchange mechanisms do not just carry waste away, they actively filter input. When cellular membranes in these microenvironments falter, they introduce noise into perception itself. This is where our consciousness meets the veil of Maya, a perceptual illusion generated not by faulty thought, but by biological friction and chemical haze between cells. If your reality can be an elegant metabolic error, then who or what designed that filter? So the filter we discuss is not an external imposition. It is a local chemical dialogue between your cells themselves. This moves us past mere observation into active authorship of awareness. If metabolic error defines reality sometimes, then optimizing that environment allows you to tune the instrument itself. The key is cultivating what I call perivascular purity, ensuring those microspaces around your vessels maintain peak exchange kinetics like a perfectly tuned filtration system in an advanced distillery. It demands not just rest, but dynamic input. It requires treating these interstitial spaces as active regulators of subjective experience. Start by demanding deep periods of cellular quiet to facilitate efficient waste clearing and allow the inner architect, the glial network, to restore optimal structural harmony. The conscious effort is your most direct intervention into integrated cognition. The full research archive is at jmtomusofficial.com. What is our final verdict? Integrated awareness is not some mystical state. It is a measurable biological rhythm achievable through micro-environmental perfection. We have seen how subtle failures in interstitial exchange generate perceptual noise. The true breakthrough lies here. We can treat your cortex not as an endpoint, but as a tuning instrument itself. Instead of just hoping for clarity after stress, you must intentionally design the conditions for peak metabolic synchrony, like calibrating a high-precision clock. Focus on achieving flow states through precise input regulation that minimizes local chemical friction between cells. This intentional refinement is how we transition from merely stabilizing reality to actively authoring it. The full research archive is at jmtomusofficial.com.